Good morning. This is Michael Dong on episode number 8, Why UV Detection is Mandatory in Pharmaceutical Quality Control. Scope. It turned out that reverse phase chromatography, or RPC, with UV detection dominates in pharmaceutical analysis. And it turned out also that a photodiode detector, or PDA, is the default detector in most pharmaceutical labs. The question is, why is a simple UV detector and not the omnipotent mass spec that dominate in pharmaceutical analysis and also in QC or quality control? Here are a few reasons. First of all, most drugs have chromophores and absorb light in the UV invisible range. This is due to the presence of structural moieties such as aromatic rings, conjugated double bonds, carbonyl groups, nitro or heteroatom systems in the drug molecules. The chromophore exhibit light absorption due to electronic transitions such as the pi to pi anti pi orbitals or the non-bonding orbital to anti-pi orbitals. It turned out that UV detector is a good fit for QC. First, relatively inexpensive. Second, it's very easy to use and highly reliable, particularly with annual PM or preventive maintenance. Great sensitivity with a typical noise of plus or minus 1 times 10 to the minus 5th AU, meaning picogram to nanogram detection. And when monitoring at the lambda max of the API of the main component in the drug substance drug products, it is highly specific to the active pharmaceutical ingredient and also its related substances such as process impurity or degradation products. Also, highly convenient, meaning that it can support a single point calibration. This is because UV detectors typically have 5 orders of magnitudes of linearity from LOQ, limit of quantitation, typically about 0.05% of the drug substance sample, to linear to 1.5 AU or absorbance unit. In early drug development, the area underneath the curve, HPLC area percentage, is generally regarded as the same equivalent to the percentage of weight percent. Now, why indeed is UV detection mandatory or required? The big question is actually precision, because UV detection have precision much less than 0.5% RSD, which is actually required for release testing of drug substances with a typical specification of 98.0% to 102.0%. Now, routinely, an RSD of about 0.2% is routinely achievable in UV detection with modern autosamplers. This is because in UV detection, all sample analyzed flow through the UV flow cell. And also, UV detection generally less susceptible to matrix effect, unlike those of mass spec or CAD. Next question is, how about mass spec or CAD detection? For the few non-chromophoric drugs which are very infrequent, the alternatives are a sample digitization, which is really not a very good idea because of the lack of precision and also multiple derealization product being formed. Generally, there's two detector which is being considered. One is a mass spec, particularly maybe a single point mass spec, a single quad mass spec, or a CAD standing for charge aerosol detector. Nevertheless, both mass spec and CAD are much less precise than UV detection, typically about 1 to 5% RSD, which is not really good enough for release testing in QC. And this is because both of these detectors, the analyte undergoes nebulization, desolvation, get rid of solvent, and also go through the ionization process. For mass spec, only a small fraction of the analyte actually enter the mass spec, and typically an isotopic internal standard is needed for quantification. There are also very prominent matrix effects such as ionic suppression or ionic enhancement, and also generally multi-level calibration are typical. However, there are also limitations for UV detection. For instance, in a stability indicating assay, a UV detector requires baseline resolution of all impurity index product from the API and from each other for accurate quantification in regulated testing under GMP. Also, qualified reference standards are needed to quantitate and also for identification of unknown peaks. However, this particular limitation can be mitigated by incorporating mass spec during HPLC method development during early development. In summary, why UV detection is required in pharmaceutical analysis? Because UV detection and pharma analysis are good fits. Because most drugs are chromophoric. Also, the detector, UV detector is inexpensive, reliable, both specific and sensitive for related substances. It supports single point calibration, making it very convenient without the need to go to multi-level nonlinear calibration. Finally, a performance for precision less than 0.5% RSD is really required for small molecule drug substance release testing for the generation of CFAs, standing for Certificate of Analysis. Here are some references from the HPLC and UHPLC for Practicing Scientists, 2nd edition, Chapter 4. This particular white paper published in 2019 on ultraviolet detectors, perspective, principle, and practices, and also a YouTube channel, HPLC for Practicing Scientists, particularly in episode number 16. In episode number 8, we discuss why UV detections in pharmaceutical analysis QC is required and why mass spec and CAD might not have sufficient precision performance to support quality control. On that note, I'd like to thank you for your attention, and we see you next time.